Did you know that the World Health Organization identifies tuberculosis as one of the top 10 causes of death worldwide, and of infectious diseases, it causes the most mortality? While its prevalence has declined in developed countries such as the United States, tuberculosis, or TB, still remains a public health threat for those in developing countries. Although the introduction of antibiotics like streptomycin and isoniazid in the mid-20th century revolutionized TB treatment, the emergence of drug-resistant strains has created new therapeutic challenges. Hi, I'm Krishna Sulibhavi, and today I'm going to be presenting about Mycobacterium tuberculosis, current diagnosis and treatment, along with the possibility of bacteriophage therapy. Mycobacterium tuberculosis, or MTB, the bacteria that causes tuberculosis, possesses several unique structural and biochemical traits. The bacterium is rod-shaped, non-motile, catalase positive, and is an aerobic bacteria. MTB is an acid-fast bacteria due to mycolic acid that resides on its surface. This acid acts as a protective layer around the bacteria, preventing it from being gram-stained. These special characteristics are vital for MTB survival in the human body. Mycobacterium tuberculosis has many strategies to thrive in the human body. MTB typically spreads to the lungs through inhalation of contaminated aerosol drops. After reaching the alveoli located in the lungs, the bacteria is usually detected by macrophages that guard the lungs, but infections of alveolar epithelial 2 cells can happen before this. In addition to the lungs, the most common place for infection are the lymph nodes. When alveolar macrophages detect MTB as a threat, they phagocytose or engulf the bacteria with their cell membrane. Then, the macrophage attempts to lyse the bacteria through phagosome maturation, but the bacterium interferes with this process. This results in the bacterium being able to infect the macrophage. As more and more macrophages begin to get infected, other immune cells such as neutrophils and lymphocytes get sent to the site of infection. These immune cells start closing in on the infected macrophages and start forming a granuloma to prevent the bacteria from escaping and causing disease. A tuberculosis granuloma is made of neutrophils, natural killer cells, dendritic cells, and more. A granuloma could stay dormant and not rupture your entire life. A tuberculosis granuloma rupturing could be caused by immunocompromising conditions such as HIV or malnutrition. If the granuloma ruptures, MTB begins infecting more and more cells, causing symptoms that include a persistent cough, high fevers, hemoptysis, which is coughing up mucus, and sweating in the night. If tuberculosis is not treated, it may eventually lead to death. This is a visual representation of MTB's disease pathogenesis, starting with the host inhaling the disease, inhaling the bacteria, and then progressing all the way to when the granuloma ruptures and the symptomatic stage begins. Diagnosis of a tuberculosis includes 2 types of tests: triage test and confirmatory tests. Triage tests are used to determine if the patient has a high probability of having tuberculosis, and confirmatory tests confirm this. Some triage tests include screening symptoms, chest x-rays, CRP blood tests, cytokine tests, and cough or chest sounds. Positive triage tests for a symptom screen would be having hemoptysis. A positive triage test for a chest x-ray would be granulomas being found. Positive triage test for a cytokine test would be specific inflammation markers, and a positive triage test for a respiratory sound test would be crackles in the sounds. On the other hand, confirmatory tests consist of nucleic acid amplification tests, or NAATS, to detect the DNA of tuberculosis. And the other type of confirmatory test is the sputum culture method. The gold standard for diagnosis is the sputum smear culture test, which can take up to 1 to 8 weeks. A sputum smear culture test consists of analyzing the substance for bacteria respective to tuberculosis that is coughed up from the lungs of possible tuberculosis patients. The reason that this method isn't as widely used as it can be is because of the time it takes to provide results. NAATs are much faster and are not as accurate, but they usually are pretty accurate. After a positive diagnosis, the proper treatment using antibiotics must be administered in order for a fast recovery for the patient. There are 5 main antibiotics used to combat MTB: streptomycin, isoniazid, rifampicin, pyrazinamide, and ethambutol. Streptomycin is the earliest TB drug and was created in 1944, and it was one of the greatest breakthroughs in human history. Isoniazid is a drug that inhibits an important enzyme for the bacteria called the catalase peroxide enzyme. Rifampicins inhibit gene transcription by binding to the bacterial RNA polymerase. Pyrazinamide kills bacteria inside the granuloma, and ethambutol inhibits synthesis of important parts of the MTB cell wall. Although these antibiotics are usually successful in eliminating the pathogen MTB from the lungs, there are further complications if the bacteria present as drug resistant. Bacteria that are drug resistant are unable to be eliminated by certain kinds of drugs or certain kinds of antibiotics. Most of the time, MTB is not resistant, but there are also cases where the bacteria slowly evolves to avoid certain drugs. There are 3 main different drug-resistant forms of MTB: multi-drug resistant TB, extensively drug-resistant TB, and totally drug-resistant TB. Multi-drug resistant TB is resistant to 2 first-line drugs. Extensively drug-resistant TB is resistant to 2 first-line and 1 second-line drug, and totally drug-resistant TB is resistant to all known TB drugs. A first-line drug is a drug that is commonly used and if a patient has no allergies or complications. A second-line drug is used if a patient has a possible allergy, or in this case, the bacteria is drug-resistant to a first-line drug. Due to the problem of drug resistance, some individuals are contemplating the use of alternative treatments that can include bacteriophages. To start off, an overview of bacteriophages. Bacteriophages are viruses that specifically infect and replicate within bacterial hosts. Their structures include a head and a nucleic acid core that is surrounded by a protein capsid. There are 2 main types of phages: temperate phages and virulent phages. Temperate phages can either go into the lytic or lysogenic phage, and virulent phages that are phages that just move into the lytic stage. The lytic stage starts with the phage injecting its own DNA into the bacteria and circularizing this DNA along with the bacterial genome. After this, the phage begins to replicate and finally lyses the bacteria. In contrast, during the lysogenic stage, the phage integrates its DNA into the bacterial genome and replicates passively as the bacterium divides. This integrated viral DNA, called a prophage, is copied each time the bacterial cell divides. In short, the lytic cycle is when a bacteriophage enters a bacteria, replicates, and explodes the bacteria, and the lysogenic cycle is where the bacteria puts some of its own DNA in the bacteria DNA so that it replicates every time the bacteria replicates. The first recorded patient with tuberculosis to be cured by phage therapy was a 15-year-old who had cystic fibrosis. The patient was treated with the 3-phage cocktail. A phage cocktail is a combination of phages that are able to lyse the targeted bacteria, and a phage cocktail helps protect against phage resistance, where bacteriophages are unable to be affected by phages. Bacteriophages have been shown to be affected and able to lyse M. abscess, M. ulcerans, M. avium, and M. tuberculosis. Additionally, phages have been effective against mimic granulomas in lab models with M. smegmatis as the host. In order to deliver the phages into the lungs of an infected human, the phage must be inhaled or injected into the bloodstream. In order to lyse all the bacteria present in the body, the phages often require a carrier bacterium. A carrier is typically a bacterium of the same family as a target bacteria but does not cause a disease, an example being Mycobacterium smegmatis. Using a carrier enables the number of bacteriophages to stay high enough for all of the Mycobacterium to be eliminated. This diagram here shows how Mycobacterium smegmatis is able to be used in eliminating the disease-causing Mycobacterium tuberculosis. Although phages have shown promise in treating patients, there are many challenges to phage therapy regarding tuberculosis. Some challenges for phage therapy regarding tuberculosis include bacteria developing resistance to the phages that infect them, and the limited host spectrum where phages are not able to infect enough types of bacteria. Additionally, there is the question of antibody resistance when it comes to delivering the phages into the body. In at least one case study, a patient has produced an antibody against the bacteriophages, rendering the bacteriophage useless. Overall, tuberculosis can be treated with a variety of drugs, but due to the new drug resistance, bacteriophages are being considered. Bacteriophages are still in new phases of development, with antibiotic treatment being much more viable under most circumstances. As drug resistance rises in the world, knowing more about alternate methods of treatment will be vital for global health, especially because MTB's deadly disease pathway through the lungs makes it vital to learn more about. Also, extra research is needed in the field of bacteriophage therapy. Specifically, research is needed on the immune system that sometimes occurs due to a bacteriophage's immune response that sometimes occurs due to a bacteriophage's presence in the lungs. Finally, more case studies regarding bacteriophage therapy of tuberculosis are needed in order to develop more patterns and understand how the bacteriophages interact with the human body. Hi, I'm Krishna Sulibhavi, and I hope you learned something and enjoyed this presentation. Thank you.